All right. Hi, everyone. Good afternoon, and thanks for coming. My name is Michael Owens. I'm a fifth-year PhD candidate in the Biochemistry and Molecular Biophysics graduate program here at the University of Pennsylvania, and I'll be your moderator today. We have two great speakers lined up for you, but before I introduce them, I'd like to briefly introduce all of you to the university's recently launched Institute for RNA Innovation. Under the leadership of our director, Dr. Drew Wiseman, the Institute brings together researchers from a wide array of disciplines across UPenn to lead the way in the study of basic RNA science, the development of RNA-based vaccines and therapeutics, and the development and targeting of lipid nanoparticles and other delivery mechanisms. Our first speaker this afternoon is Dr. Lijun Zhou, who is an incoming assistant professor in the Department of Biochemistry and Biophysics in the Institute for RNA Innovation. Dr. Zhou received both her bachelor's degree in biology and her PhD in biochemistry and structural biology from Tsinghua University in Beijing. During her PhD in the lab of Yigong Shi, she was the lead author on several crystallographic studies of the yeast LSM 1-7 RNA degradation complex in the LSM 2-8 mRNA splicing complex. After her incredibly productive PhD, Lijun joined the lab of Jack Szostak at Harvard for her postdoc, where she's built up a sizable body of literature regarding how the chemistry of early Earth may have facilitated the formation of the first RNA molecules. When her lab opens at Penn officially next January, Dr. Zhou plans to expand upon her research into the RNA world in some exciting ways that she'll be telling us about today. Please join me in welcoming Dr. Lijun Zhou.
Okay, thank you very much for your kind introduction. And today is my great honor to be here to share about my postdoctoral work in the past few years that have been done in Jack Szostak lab at Harvard University. And I have been trained as a biochemist and chemical biologist. So I try to work at the interface of chemistry and biology to understand how life begins in the RNA world hypothesis. And today I'm talking about how we employ non-natural nucleic acid to understand the RNA world. And when we really look up in the sky, it's like very hard to imagine that like how life begins in this universe. When we look around us on this earth, we see various forms of life, and we biologists try to work on different problems to understand each pieces of puzzle about life. And ultimate puzzle is that how life begins. And to understand this problem, we need to work on the chemistry of these biomolecules, small molecules that can build up the life. And this is the timeline of history of life on Earth. And you can see that it has been more than 4 billion years ago that Earth began. And it took around less than 0.4 billion of years for the RNA world to have the first type of the RNA form life. And actually, it's relatively short of time when we think about it, looking back in the whole history. Because after we have the first type of DNA protein life, it takes almost 2 billion years until we have the first eukaryotic cell. And then it takes another 1 billion years from single cell life to multiple cells life. And you think about that, then 0.4 billion years timeline is relatively short. So this type of chemical that happen and improve into life should be able to be mimicked and understand inside the lab. And here is like 3 different components of nucleotide that we talk about today. So we try to use chemistry to modify these 3 different components, including living group, sugars, and nucleobase, and employ this to understand how the RNA self-copying can happen and RNA self-assembly. And this is the simple talking about the genetic information copying process. In the modern world, we have the DNA polymerase and use the DNTP, and this is very fast and efficient. Everyone here can do the PCR reaction very efficiently. You can copy 1,000 base pair of DNA within a minute inside the PCR tube. And in the RNA world, presumably, we have the RNA polymerase ribozyme, which is pretty big and long RNA that fall into tertiary structure. It might be longer than 100 or 200 nucleotides. It can form into tertiary complex structure and uptake this kind of small RNTP and use it to copy on the RNA template. However, in the pre-RNA world, which is in the middle between when the Earth just began and we have the small molecule coming back, try to build into the small building block, but it have not evolved into such a long RNA that like 100 nucleotides, sophisticated structure. In this pre-RNA world, we don't have the ribozyme yet because the ribozyme is too big, 100 or 200 nucleotide long. We only have the small molecule and small short pieces of oligonucleotide floating around. Then at this point, how can RNA information can be copied by itself without the help of enzyme? This is something that we try to understand. And we found that by modifying the living group, we can accomplish the RNA self-copying without the help of enzyme. This is the video that shows how enzyme-free RNA copying might happen. Presumably, we have an RNA template, which is a single-strand RNA, and those mononucleotides can base pairing to the template with Watson-Crick base pair. Then the hydroxyl group of the 3' at the upstream mononucleotide can attack the 5' phosphate of the downstream oligonucleotide, and then the phosphodiester bond can happen and form. And in this process, you can see that it requires the self-base pairing and very good SN2 reaction to happen. So this is quite problematic and difficult to happen without the help of enzyme. And inside the lab, we found that we use this kind of experiment to understand this process. We call non-enzymatic primer extension with the primer and template duplex formation. And there is an open reading frame here that single strand template region. And then we add activated monomer that we modify 5' end of it with imidazolium group onto it. And then at each time point when we take the sample and run the gel, we can see that that 3' end of primer keep extended and grows. And then we can calculate the reaction rate and see this reaction. And this work can go back to 1968 in Leslie Orgel's lab, when he found that if we use 5' imidazolium in mononucleotide, we can see the primer extension happen without the help of enzyme. But at that time, the reaction is so slow that it will take over than a week to see 25% of primer got extended. And later on, we found that by modifying this imidazolium group, we can get a better reaction rate. And in the recent years, in the short stack lab, we found that if we use 2-amino imidazolium with the help of helper, we can accomplish the copy up to 7 nucleotide or 10 nucleotide long in this range within a day. So this is relatively fast when we think about the process of the whole evolution. So we solved the problem of how prebiotic chemistry small molecule build up into the small short oligonucleotide in the pre-RNA world. However, we still don't know how this pre-RNA world evolved into RNA world, because this 7 to 10 nucleotide long is not long enough to perform sophisticated machinery at ribozyme to help the RNA self-copying in the RNA world that needed. So in this 0.2 billion years, something happened that these small short pieces oligonucleotide can self-assemble and evolve in Darwinian evolution, and then the RNA will begin. So the problem to solve with this problem first is that the difficulty to copy long RNA template. And it's very simple, intuitive to think about, can we use ligation reaction to substitute polymerization? Excuse me. Because the polymerization, you can add only one mononucleotide at a time. But in the ligation reaction, you add pieces of short oligonucleotide at a time. So when you try to copy the template with the same length, you need fewer steps of reaction with ligations. However, when we try to do this type of non-enzymatic RNA ligation reaction, we found that the reaction rate is extremely slow. You can see in this that the best rate that we can get in this scenario is 0.02 per hour. This is almost 500-fold slower than an enzymatic RNA polymerization. And if you look at the presentative gel, you can see that after 4 hours of reaction, we have only fainted band of ligated product, which is less than 10%. And it will take over days for the primer to be totally extended by non-enzymatic ligation. You may think that, oh, we have billions of years for evolution, so it's fine with this rate. However, if you think about it, we need the ligation reaction to be faster than the RNA hydrolysis rate. Otherwise, we won't be able to accumulate any meaningful copying product. So in this case, this kind of reaction rate is too slow to do any meaningful RNA copying. So we try to understand why the RNA ligation reaction is so slow, and we found that the mechanism of the RNA polymerization and ligation is totally different. In the non-enzymatic polymerization reaction scenarios, two mononucleotide monomer can react with each other and form this type of bridge intermediate structure with the imidazolium group in the middle. And then it's not mononucleotide, it acts as a dimer that binds at the downstream of the template, and then one mononucleotide together with imidazolium group act as a leaving group. And this leaving group is much stronger than imidazolium leaving group by itself. So that's why non-enzymatic polymerization is much efficient compared to ligation. So we asked if we can use an enzymatic ligation to do something meaningful to overcome the problem that we can only copy the short template. We need to ask back and see, can we make this 3' end of 3' hydroxyl group of ribonucleotide to be much better protonated during the reaction. And in the prebiotic Earth, presumably at that time, it might be peptide or other small molecule that co-evolution together with the RNA that help to deprotonate 3' hydroxyl group of RNA when it tries to copy itself. However, inside the lab right now, we still haven't discovered those type of peptide or catalysts that prebiotic enough and can help with this type of reaction yet. However, we can use non-natural nucleic acid with the sugar modification, which have much better nucleophile at 2' end to mimic the behavior of RNA and use it as a proxy of RNA to understand how the RNA assembly can happen if we can deprotonate the 3' end of the primer in much more efficient way. And here, we use this 3' amino dideoxyribonucleotide because the amino group here is a much better nucleophile compared to the hydroxyl group here. And it's very interesting that like when it forms polymer, this 3' mpDNA, it likes to form A-form duplex just like RNA, and it can bind to the RNA very strongly. So this behavior makes it very interesting proxy that we use and employ it in the origin of life and RNA we're exploring. And we hope that in the future, we can use this type of non-natural nucleic acid to understand another biology question inside the in vitro assay. And here we come back and examine this reaction again. When we substitute the 3' end of RNA primer with this type of 3' amino mononucleotide, we found that the reaction rate, the ligation rate can be increased for almost 50-fold. And right now, within 15 minutes, we can see the accumulation of pretty good amount of the ligation product. However, this reaction rate is still not good enough because we would like to see it can go up to around 10 or 20 per hour, because in that case, it would be comparable with non-enzymatic polymerization, and we can use it to make some meaningful functional reaction. So we asked if we can optimize this reaction with a better leaving group. And here we employ the hydroxy ethylene imidazole, HEI, within the reaction. This is very reactive imidazolium. It's so reactive that we cannot purify HEI activated tetramer to do the ligator for the ligation reaction. But what we can do is that we can add the HEI within the solution of reaction, and it can happen in situ, the rapid exchange between the imidazolium group, and we can see the reaction right away. And we found that when we add HEI in the solution of the reaction, we can see another tenfold increasing in the reaction rate. And right now, the reaction rate of non-enzymatic ligation reaction can go up to something between 17 per hour to 18 per hour. And this reaction rate is very rapid. And we found that like this is very encouraging to use this type of reaction to mimic the formation of long oligonucleotides. And you can see in this representative gel that within the 5 minutes of the reaction, you can see most of the primer got ligated. Excuse me. So here, since we found a rapid way to conduct non-enzymatic RNA ligation, we asked if we can copy long oligo template. And in this case, for proof of concept, we prepare a template of 32 nucleotide long, so which is much longer than what we can accomplish by monomer addition polymerization. And in this case, we add the tetramer into it. So it requires 8 steps of the non-enzymatic ligation until the full-length product to happen. And we found that like within an hour, we can see most of the primer got extended and form the final full-length product. So in this case, as a proof of concept, we show that by multiple steps of non-enzymatic RNA ligation, we can achieve the copying of long RNA template efficiently within hours. So it seems like we have solved the problem of how to copy the long RNA template. Then the next question happens is that like how the very first long RNA template begin in the RNA world. Because in the pre-RNA world, we only have the short oligonucleotide floating around, how we can have the long RNA template happen right at the beginning. And here we come to the very simple concept again, because when we try to understand origin of life, we want things to happen in prebiotic condition that's achievable just by the physical and chemical properties of oligonucleotide by itself. And we found that like if we can, and we see that like if we can use the concept of splinted template that only partially base pairing to each other, we can get the product that is much longer than the starting material by itself. Excuse me. So as a proof of concept, we start by the tetramer assembly. We use very short tetramer CUGA that ended by 3' amino group, and 5' end of it is modified by imidazolium group. And we also use another tetramer as the splinted template ADUC. And you can see that between the tetramer and ligator, there are only 2 base pairing here, which is extremely weak. But in this case, it's very extremely conditioned to see how by employing only 2 base pairing can we see the formation of long oligonucleotide efficiently. And to monitor this reaction, we also employ the primer duplex with 5' are labeled by fluorescence and run the gels. And long story short, we found that in optimized condition, we can see the growing up to over 100 nucleotide long product starting from tetramer within a day. So in this case, we have demonstrated the possibility to grow into a very long RNA template just starting from tetramer. So this kind of RNA self-assembly is very efficient and very simple, and it sounds prebiotic possible that can happen in the prebiotic earth. Similarly, by employing this kind of non-natural nucleic acid, we have demonstrated the possibility, the formation of long RNA copying and the emergence of long RNA template. But what next question that like we not only want to get long oligonucleotide, we want to get something functional. So with this method, can we assemble a functional ribozyme? And here is the shortest RNA ligase ribozyme that ever reports. It's only 52 nucleotide long. And when it falls into second sphere structure like this, it's able to ligate the substrate, which is 40 nucleotide long 3' 5-phosphate to the 3' end of the cell. So in this case, we try to see if we can use non-enzymatic RNA ligation reaction to assemble this kind of RNA ligase ribozyme, and we hope that it can be functional. And because this ligase ribozyme is about 52 nucleotide long, so we try to assemble it with 5 pieces of short oligonucleotide, 12 nucleotide long, 10, 10, 10, 10. And we hope that with the ligation reaction together, we can get this 52 nucleotide long, and then hope that it will be functional. We think that 10 nucleotide long is something is reasonable in the pre-RNA world, because with this length, it's possible to copy by itself and it's not too long, so that like RNA separation might be possible. And here we start by this ligation reaction with very straightforward. The splinted that we designed for the first time have 5 base pairing on the upstream and another 5 base pairing with the downstream. And we try to put these 5 pieces ligator together. And this non-enzymatic ligation reaction is really efficient. So within a day, we can see that most of the ligator ligate together, and we get the full-length product of 52 nucleotide long. Then we add the substrate into the system. We heat up everything to 75 or 85 degrees Celsius, and then cool it down to 48 degrees, which is the optimum temperature of this type of ligase ribozyme. But we did not observe any ribozyme ligase activity at all. So we come back and see what happened during this reaction. This is the theme of how we design this experiment. And this is the problem that we call strand inhibition, which is quite famous problem in prebiotic study when we try to understand origin of life. So what actually happened is that after non-enzymatic ligation reaction happened, we heat up everything and we try to cool it down. We attempt to see the folding of this type of ribozyme and the dissociation of template. However, this template by itself can bind to the non-enzymatic ligation product very strongly, and it inhibits the folding of the RNA ribozyme by itself. So what happens is that it will stay in these configurations that the templates still bind to the non-enzymatic ligation product and found a duplex that cannot fold into tertiary structure and conduct its function. So this will inhibit the enzymatic ligation reaction later on. So how to solve this problem? Like we go back and see that we try to modify living group, we try to modify the sugar, and this part we see we ask if we can do some prebiotic possible modification on nucleobase to help to lower this type of the RNA self-base pairing, but still maintain pretty good specificity of the template and the ligation product. So we think about two different solutions by substitute GC base pairing either to inosine C base pairing or GU wobble pairing. Inosine is very prevalent in the modern biologies, that it is very common to see this kind of modification in cells nowadays. And it is also very prebiotic possible in prebiotic condition to synthesize inosine as well. And we found that inosine might be a good substitution of guanosine in the prebiotic earth. And GU wobble pair is something that very unique in the RNA structure. So it's different from DNA that like GT pair might not happen. We always see GC pair and AT pair in DNA. However, in RNA, GU wobble pair is quite specific and it's common to see. And these two type base pairing have only 2 hydrogen bonds, so it can be much weaker than the GC pairing. So we go back and look at our splints template, and we found that we have quite a lot amount of GC base pairing. So we substitute some GC base pairing with GU wobble pair. And we found that with this substitution, the binding affinity between ligator and splint is a little bit weaker. So the non-enzymatic ligation reaction is a little bit slower, but we still see the majority of full-length product form within 1 or 2 days. And then when we add the substrate, heat it up and cool it down, refolding everything, for the first time we see the ribozyme ligase activity that like 3' it can ligate the prebiotic substrate into the 3' of it. And again, we also test the hypothesis about inosine and cytosine, and we substitute a few guanosine in the splint with inosine so that we can have only 2 hydrogen bonds. And we found that like the same thing happened. We can see slightly slower non-enzymatic ligation reaction, but we can see relatively efficient ribozyme ligase activity test. So in this case, we have shown and demonstrated the formation, the assembly of a functional ribozyme started from very short oligonucleotide. And this might be a demonstration about what might happen between the transition from pre-RNA world to the RNA world. So at that point, because the copying of long oligonucleotide is very difficult, so even though the protocell might require some ribozyme that is 50 or 100 nucleotide long to conduct some meaningful function to maintain its life, but it's very hard to copy this length of the primitive genomes. So what might happen that it carry a set of short oligonucleotide as a genome, like in this case, might be something around 10 nucleotide long, so that it can copying each pieces of 10 nucleotide long relatively efficiently. And then when it's required, it can assemble and use non-enzymatic ligation reaction to form big and functional ribozyme. And even though by canonical RNA splinted, it might be some problem, it can overcome by using another set of template that was copied independently, but simultaneously with certain modification on the nuclear base and introduce some GU wobble pair or inosine cytosine pairing to lower the melting temperature. And in this case, we can see the efficient copying of the short oligonucleotide cells, and together it can assemble into functional ribozyme when needed. And with this, I would like to thank Jack Shostak and all the lab members at Harvard University to help me to be a fantastic family to work on this topic together. And I'm really much looking forward to start working with a group of people at Penn Institution of RNA Innovation starting in January. And we are hiring. If anyone interested in RNA chemistry or XNA selection and RNA therapeutics topic, please feel free to contact me. And if you have any problem regarding with my talk today that I don't have a chance to reply right away, please feel free to email me. And thank you very much for your attention, and I'm happy to answer any questions.
Thank you. That was fantastic. Looking forward to being your neighbor in a month. While we're waiting on some questions to come in the Q&A, as a reminder, please post your questions in the Q&A. If you have them, if you'll indulge me in a quick question, just in general about doing work, speculating, and trying to do things with the pre-RNA world, how careful do you have to be about trying to match the conditions of that kind of prebiotic Earth? Are you doing everything in a glove box to keep oxygen out? Are you caring about different temperatures, metal oxidation states? How much does this keep you up at night, I guess?
Thank you very much. This is a very good question. So I think in general, right now, we try to work things at pH 8.0 and with high magnesium. So we can just do it on the bench because it's not much difference between inside glove box and on the bench. But there's some certain conditions, like, for example, when we try to study the effect of other metal ions, such as iron, on this type of reaction, we will need to use the glove box because that, like, in that case, it can be huge difference, right, between using it just on the air and inside the glove box. But otherwise, like, we don't need to do it like that every single time. If we know that that type of reaction cannot generate different or too much difference between these two conditions.
All right, thank you. I think we have one or two questions currently being typed in the chat. So we'll, I think, wait another minute or so for everyone to type in their questions.
I will be available to answer in the Q&A box during the talk of Robert Wang.
Sure.
Thank you.
All right. Let's see. Oh, one from Sarja Dasgupta. What do you think is the biggest problem with the RNA world? I'm not, perhaps you could to add a clarification of that. I'm not entirely sure what you mean by that. I'm not sure, do you know?
Thank you very much for this question. And I think it's the big question by itself. I think the biggest problem to explore the RNA world hypothesis, but I did not have time to talk about today in my talk, is how to coupling the process of the cell membrane growth together with the growth of the RNA by itself. Because these two independent problems have been investigated extensively in the past few decades, that like how the cell membrane can grow and how the RNA can grow without the help of enzymes. However, to coupling these two processes together and allow the copying and the daughter of RNA to be dissociated precisely within two daughter cells, this is very difficult. And I think that will require a while to investigate, to understand how this process can happen without the help of enzymes. Thank you.
All right. In the interest of time, I think we're going to switch over to our next speaker. If you have questions for Dr. Zhou, please feel free to add them into Q&A. And she'll be around kind of answering the background. So let's thank Dr. Zhou again. And then while he sets up, our next speaker is Robert Wang, who is a PhD candidate in our Genomics and Computational Biology program at Penn. Robert earned his bachelor degree in bioengineering from UC Berkeley and is currently working on his PhD in the labs of Lin and Xing, both of them in the Department of Pathology and Laboratory Medicine at the Children's Hospital of Philadelphia. Robert's thesis work has largely been focused on developing computational workflows for studying the diversity of the transcriptome across several different tissue types using long-read sequencing. He joins us today to share some of his recent work on the topic. So please join me in welcoming Robert.
Thanks, Michael, for the introduction. Let me share my slides. Let's see. Great. Okay. So again, thanks for the introduction. I am incredibly delighted to have this opportunity to represent the RNA group here at Penn. And today I want to tell you about some recent work that we've done in using long-read sequencing to study the human transcriptome. And first, I'd like to apologize in advance for my voice. Unfortunately, I'm still recovering from the flu, so my voice might be a little hoarse. Okay, so I think I'd motivate this work by a comic strip that was published nearly 10 years ago in an issue of Nature Methods. The comic starts off with a scientist shown here who is buying magazines at a magazine stand. Unfortunately, the magazines can only be purchased after they've been shredded. But thankfully, the scientist has many helpers who can help assemble these shredded pieces back into whole magazines. But the reconstructed magazines do not appear to be exactly perfect. And this is arguably how we've been doing transcriptomics for the past 10 years. Traditional methods for RNA sequencing have really served us well over the past decade and have enabled a lot of insights into gene expression and splicing. But these methods really have limitations when it comes to studying the expression of individual transcripts, which are the real functional units of the transcriptome. And so like the vendor at the magazine stand, traditional RNA sequencing involves fragmenting your transcripts into many small pieces. You can't really figure out where each piece comes from, but you need to somehow use this data to figure out whether you have, say, this transcript present or this transcript present. And if they're both present, what is their relative ratio? I'm sure that we can all agree that this seems like a very incredibly challenging problem. But on the other hand, we can now directly sequence entire transcript molecules using current long-read sequencing technologies, which have really begun to revolutionize the way we do transcriptomics. And so currently, there are two technologies that really dominate the long-read sequencing space, Pacific Biosciences or PacBio, as well as Oxford Nanopore Technologies, the latter of which I'll primarily talk about today. So shown here is a nanopore sequencer, which is small enough to actually just fit right in your back pocket. And this sequencer contains hundreds of these nanopores, which measure changes in an electric current caused by either DNA or RNA going through. And these electric signals can then be converted into nucleotide sequences. And so with nanopore sequencing, we can easily generate reads longer than 10 kilobases. And this really gives us the capacity to sequence entire transcript molecules from end to end. And so given these advantages, it's really unsurprising to now see a steady increase in the number of studies that are performing long-read RNA sequencing over the years. Yet, when we compare these numbers against the numbers of studies still performing traditional short-read RNA sequencing, we can see that there is quite a stark contrast. And so what this means is that not everyone working in transcriptomics is using long reads, despite their promising nature. And I think this really raises the question of why? What kind of barriers might be in the way of adopting long-read technologies across the broader community? And so I think the answer to this question becomes very clear when we examine the technological limitations of long-read sequencing. First, long-read sequencers have a relatively low sequencing throughput compared to their short-read counterparts. So for example, nowadays, if you have $1,000, it's very easy to generate 100 million short reads. But with the same amount of money, the number of long reads that we can get is usually orders of magnitude lower. And moreover, perhaps the more notorious limitation of long-read sequencing is its relatively high error rate. And so according to this table, which was taken from a 2019 review paper, long-read sequencing has an average error rate of around 10%. So what this means is that for every 10 bases you sequence, you would generally expect around 1 incorrect base call. I will note that these long-read technologies, in particular Oxford Nanopore, have made tremendous progress in improving their read accuracy throughout the years. And currently, there's still active progress in improving this read accuracy. But at present, long reads still remain largely error-prone. And surprisingly enough, frequent errors in long-read RNA sequencing data can actually have a rather non-trivial impact on transcript discovery and downstream analysis. And to really go about illustrating this point, here I'm showing you individual nanopore RNA sequencing reads aligned to this pretty well-known gene called CD19. And so from afar, we can see that each of these individual reads seem to cover the exons of this gene quite well. Yet when we take a closer look at these reads, we can see that these alignments around the splice sites in particular are actually quite noisy. And what's causing this is the fact that you have frequent base insertions and deletions within these long reads that can ultimately confound the process of read alignment. And so really this makes data analysis quite challenging, as it's unclear whether these spurious alignments, do these alignments actually suggest novel splice sites and thus novel transcripts, or if they're really just artifacts in our data. And so naturally, this really begs the question of whether or not we can reliably identify splice junctions from these error-prone long reads. And so to go about addressing this question, we generated nanopore RNA sequencing data for a set of synthetic spike-in RNAs, which have known structures and concentrations. And so in this context, we actually have full prior knowledge of all the splice junctions that must exist in our data. So we can readily identify alignments that correspond to correct splice junctions, as well as those corresponding to incorrect splice junctions. And so using this data, we wanted to first examine whether there are any sort of features that might explain why certain read alignments were more likely to have correct splice junctions versus incorrect ones. And what was really striking to us was that we found that splice junctions were almost always correct in read alignments where there were no mismatches, insertions, or deletions within 10 base pairs of the putative splice sites. By contrast, splice junctions tended to be a lot more incorrect in read alignments that did carry these errors near the corresponding splice sites. And so, motivated by this observation, we reasoned that this former group of read alignments, these so-called perfectly aligned reads, could potentially be used to improve both the discovery of splice junctions as well as their corresponding transcript isoforms from error-prone long reads. And so this drove us to develop Espresso, which is a new computational tool for transcript discovery and quantification using long-read RNA sequencing data. I would like to point out that much of Espresso's development was done by a very talented former postdoc in the lab, Gao. And so our tool works as follows. So it takes a set of long-read RNA sequencing alignments as input, and Espresso first identifies what we call a set of high-confidence splice junctions from the alignment data. And so these splice junctions are defined based on several layers of criteria, such as whether the splice junctions are annotated, whether they carry the consensus splice site nucleotides, or if they're supported by a sufficiently large number of these perfectly aligned reads. And so once we've identified these high confidence splice junctions from our data, we can then use them to correct other splice junctions in each of our long reads. And so to really understand how this procedure works, suppose we want to correct this second splice junction shown here. So what Espresso does is it first pulls out high confidence splice junctions that are overlapping the coordinates of the second splice junction. And then Espresso assesses how well each of these splice junctions could be used to effectively replace this putative splice junction in question. And so in this cartoon example, Espresso would consider this blue splice junction to be a better fit compared to this yellow splice junction. So the blue splice junction is ultimately chosen to be the read's second putative splice junction. Of note, this correction procedure can also recover missing splice junctions at the terminal ends of read alignments. Specifically, Espresso would look for high confidence splice junctions overlapping the ends of a given read, and then assesses whether this high confidence splice junction could then be used to be introduced into this read alignment. And so in this cartoon example, again, introducing this purple splice junction into this read alignment is favorable by Espresso, and the corresponding read alignment is updated. And so after this procedure, these corrected read alignments can then be subsequently used for transcript discovery and quantification. And so when we first developed our tool, naturally, the first question we had was, well, how well does Espresso compare to existing tools for long-read RNA sequencing analysis? And so to this end, we sought to examine the accuracy of transcript discovery and quantification for Espresso, as well as several contemporary tools based on long-read RNA sequencing data for synthetic spike in RNAs. And so these synthetic spike in RNAs can be really useful for any assessments of transcript discovery and quantification, as we have full prior knowledge of all the transcripts that are present in our sample, along with their relative concentrations. And so as you can see here on the bottom left, in terms of transcript discovery, we found that Espresso showed the highest sensitivity and precision compared to two other tools, StringTie2 and Flames. And moreover, when we examined the correlation in estimated transcript abundances reported by a given tool compared to ground truth concentrations for these spike in transcripts, we can clearly see that Espresso had the highest overall correlation compared to these 5 other tools used for comparison. And so having demonstrated Espresso's performance using synthetic spike in RNAs, we next sought to use Espresso to analyze real biological samples. And so to this end, we generated around 623 million nanopore RNA sequencing reads across a broad panel of human body tissues, representing diverse anatomical body sites. Around half of our tissues were derived from the brain, and each tissue was sequenced with a median depth of around 20 million nanopore RNA sequencing reads. And so using Espresso to analyze this rich data set, we collectively discovered around 340,000 transcripts. And as you can see here, we generally noticed that tissues with deeper sequencing tended to have more transcripts discovered. However, I think it's important to note that several of these tissues seem to deviate from this trend. For example, the testes, which had a relatively low sequencing depth, ended up having the largest number of transcripts discovered. And by contrast, skeletal muscle and pancreas, which are 2 tissues that had a relatively high sequencing depth, these tissues ended up having a relatively low number of transcript isoforms that were discovered. And when we group together the tissues in our panel based on transcript expression, we can clearly see that there seems to be a separation of tissues based on whether they are brain-derived or not. And in fact, from our data sets, we actually found multiple instances of transcripts showing brain-specific expression that might be driving this clustering pattern shown here. For example, in a gene called DNM1L, we found 2 transcript isoforms, DNM1L229 and DNM1L203, shown in green and red, respectively, that showed enriched usage in brain tissues. And so as you can see here, in non-brain tissues, the predominant isoform of this gene is this isoform DNM1L206, shown in purple. And so when we compare the structures of these isoforms, we can clearly see that they differ in combinations of exon skipping events located in 2 distal regions of this gene. And I think this really nicely highlights an advantage of long-read sequencing, because it enables us to examine how different alternative splicing patterns are cobbled together on a given transcript. And so next, I want to tell you about yet another long-read project that we pursued, where we use nanopore long-read sequencing together with espresso to profile the transcriptomes of 40 breast cancer cell lines, representing 4 distinct breast cancer subtypes. And to really motivate this project, it is becoming increasingly well known from the literature that human cancers, including breast cancer, exhibit widespread changes in alternative splicing. Yet the landscape of transcript isoforms resulting from these splicing alterations has remained largely underexplored. And so we hypothesized that a long-read-based study of cancer transcriptomes could potentially reveal novel aberrant transcripts and shed possible insights into the functional consequences of these splicing alterations that take place in cancer. And so here's a snapshot of our data. So initially, when we grouped together these cell lines that we sequenced based on transcript expression profiles, we can clearly see a separation in the samples based on breast cancer subtype. Specifically, as you can see here, cell lines representing these basal A and basal B subtypes, colored in red and yellow, appear to cluster away from cells representing luminal and HER2-enriched subtypes. And so what this suggests is that breast cancer subtypes represented by our cell lines appear to show global differences in transcript expression patterns. And to kind of get a flavor of how transcript expression patterns vary across subtypes, here's an example. So in this one gene called DNMT3B, we found an alternative transcript isoform of this gene, DNMT3B203, shown in red, that is highly expressed in these basal B breast cancer cell lines in red. And so if you look at these other cell lines, DNMT3B is predominantly expressed as this blue isoform, DNMT3B204. And so when we compare the structures of these two isoforms, we can see that there appears to be skipping of these 2 tandem exons in this basal B enriched isoform. And so naturally, this raises the question of, okay, well, what is the functional consequence of expressing this particular DNMT3B isoform in basal B cell lines? And so it turns out that these 2 isoforms of DNMT3B have actually been previously characterized, albeit not in the context of breast cancer. So for context, DNMT3B, as you may know, encodes a de novo DNA methyltransferase that plays many important roles in regulating DNA methylation during development. And so in the basal B enriched isoform of this gene, the underlying exon skipping event actually disrupts the C-terminal catalytic domain of this methyltransferase and results in a protein isoform that has been shown to be catalytically inactive. And so the fact that this catalytically inactive isoform of DNMT3B is so highly expressed in these basal B breast cancer cell lines seems to raise some very interesting questions about global DNA methylation patterns in the basal B breast cancer subtype. Moreover, from our data, we actually also found multiple instances of transcripts with elevated expression in only a single breast cancer cell line. For example, we discovered a novel transcript isoform of TP53 that is predominantly expressed in this one cell line, HCC1599. And as you can see here, in almost all other cell lines, TP53 is expressed as this blue isoform, TP53-201. And so again, when we compare the structure of these 2 isoforms, we can see that this novel transcript isoform that we discovered appears to have this retained intron that introduces a premature termination codon into transcripts of this gene TP53. And so when we begin to think about these 2 isoforms of TP53, this TP53-201 isoform, which, to remind you, is predominantly expressed in all of our other breast cancer cell lines, this isoform will produce its encoded protein, P53, which is a very well-known tumor suppressor that plays major roles in regulating the cell cycle, DNA repair, apoptosis, as well as cellular senescence. However, when we look at this novel isoform of TP53, which, to remind you, is specific to this one cell line, HCC1599, this isoform, it carries a premature termination codon and is targeted for non-cis-mediated decay. So this will not eventually be translated to make protein. And so then this raises the question of, well, why? Why is production of this novel isoform so favorable in HCC 1599 cells? And it turns out that when we analyze whole genome sequencing data generated on this particular cell line, we actually find evidence of an A to T mutation in the TP53 gene that actually disrupts a 3' splice site of this retained intron. And I find this result to be rather intriguing, since typically when we find these splice site mutations in genomic sequencing data, we instinctively think that these mutations would induce, say, exon skipping as the transcriptional phenotype. But really the consequence here of this mutation is intron retention, which has traditionally been very difficult to characterize using short-read RNA sequencing. More broadly, we actually observed that tumor suppressors in general appear to be significantly enriched for these cell line specific transcript isoforms that are targeted for degradation by NMD. And this degree of enrichment of note, did not appear to be observed for oncogenes. And so collectively, what these results may suggest is that the production of these aberrant transcripts coupled with nonsense-mediated decay could potentially represent a common mechanism for inactivating tumor suppressors in individual tumors. And so to summarize, I think we're now sitting really at this precipice of a revolution in transcriptomics, where long-read technologies are slowly enabling us to directly sequence full-length transcript molecules, providing a more complete view of the transcriptome. However, currently, one of the major challenges of analyzing long-read RNA sequencing data is the relatively high error rate of long reads. These long reads really pose a major challenge to any downstream analysis that we do. And so to really address this critical barrier, we developed a new computational tool, Espresso, that can robustly discover and quantify transcript isoforms using error-prone long-read RNA sequencing data. Our tool is now publicly available for download from the following GitHub link. And collectively, across the works that I presented today, we generated and analyzed nearly 1 billion nanopore RNA sequencing reads, covering multiple human tissues and cancer cell lines. And from these analyses, we were able to uncover multiple transcript isoforms with potential relevance to both human health and disease. And so with that, I would like to acknowledge the individuals from both the Lin and Xing labs who helped contribute to this work. Thank you all for listening, and I'd be happy to take any questions.
Thank you, Robert. That was really interesting. So we have one question in the Q&A so far from an anonymous attendee. They ask, do you have a benchmark library to assess if the synthesized 68 spike-in RNAs introduce bias or noise into new sequencing data?
Oh, sorry. Could you repeat the question?
Oh,
I
sure.
ended up, having to cancel the screen share.
Sure.
I heard about the spike-in RNAs. Yeah.
Do you have a benchmark library to assess if the synthesized 68 spike-in RNAs introduce bias or noise into new sequencing data?
Oh, yeah, that's a great question. So actually, we didn't synthesize these spike-in RNAs ourselves. These are actually commercially purchased from a company called Lijun, and they produce these spike-in RNA standards for a wide variety of analyses, including benchmark analyses for gene expression, as well as transcript isoform analysis. And these have been widely used. Actually, these standards, are actually designed to test possible biases in these analyses. For example, these spike-in transcripts were synthesized to have varying concentrations as well as varying lengths. So having all these different covariates be present in these spike-in transcripts allows you to effectively test potential biases in, discovering transcripts based off of length bias as well as concentration bias.
Okay, thank you. So two more questions popped up. One from Junji Go. He says, great talk. Wouldn't the isoforms you talked about be equally detectable by short reads?
Yes, that's a very good question. So to kind of, to answer this question, really have to think about, okay, well, there are some transcript isoforms out there, for example, actually, I think to better contextualize this, let me just share my slides again. Right, so can you see my screen? So, there are definitely some transcript isoforms out there that might not be easy to characterize using short-read RNA sequencing data. For example, these transcript isoforms that we discovered using DNM1L, these different transcript isoforms involve different combinations of alternative splicing events located in distal regions of the gene. And with short reads, however, these short reads are simply just not long enough to reveal how these different alternative splicing events are coupled together on a given transcript. And so therefore, detecting and quantifying these transcripts, determining their relative abundance using short reads is incredibly difficult and potentially error prone. However, with long reads, we can directly sequence the entire transcript molecule. And so how these different alternative splicing events located in distal regions of this gene, how they're coupled together, you lose that ambiguity with long reads.
All right, thank you. Two more questions had popped up. One from Billy Schleif. Does Espresso work with native RNA reads?
Yes. Actually, when we performed our benchmark evaluations of Espresso, we actually evaluated Espresso using multiple different protocols for the nanopore RNA sequencing, including the direct RNA sequencing protocol. When we did our benchmark evaluations, we showed that for both native RNA sequencing data, as well as the very standard cDNA sequencing nanopore sequencing protocol, Espresso outperforms almost all the contemporary tools that we use for our benchmark evaluation.
All right, and then what's looking like it? One last question from Boris Zinstein, excuse me. In the case of the nonsense-mediated decay targeted tumor suppressor RNAs, you seem to see a lot of the RNA. Do you have evidence that that they are really being degraded? Could these cancer cells make truncated dominant negative versions of the protein?
Yeah, that's a really great question. I actually don't have the slide for this. But when I showed you the relative abundances of those transcripts, for example, the TP53 transcript was targeted for nonsense-mediated decay, what I showed you were actually the relative proportions of that transcript. What I didn't show you were the actual abundances on the level, like the number of normalized copies. And so when we actually found that total expression of TP53 transcripts in that particular cell line that was enriched for that NMD targeted transcript, overall TP53 expression was actually very low. And we also have further evidence that this transcript is NMD targeted. We actually performed a UPF1 siRNA knockdown in that particular cell line, and that we found evidence that that particular NMD transcript had elevated levels.
All right, and a couple more. Stefan Muljo asked, since in general you have fewer ONT reads, how confident can you be in the quantification of the isoforms?
Yeah, definitely. So actually in our paper for Espresso, we performed various benchmark evaluations for, what is the minimum number of nanopore sequencing reads that we need to reliably quantify transcript isoforms. And, from our benchmark analyses, we found that at least 5 million long reads are needed to reliably quantify these transcript isoforms. And actually all the analyses that we've showed you today, those samples were sequenced with over 10 million long reads. And, that is sufficiently deep enough to have a reliable quantification for these transcript isoforms.
All right, and that brings us to just about 5 o'clock on the dot. So I think we'll call it there. There are no more questions in the Q&A. So please join me again in thanking our speakers. Thank you all for your attendance today, and happy holidays.